My project is called FAABulous, which is an abbreviation for the title Future Arctic Algae Blooms and their role in the context of climate change. Algae are really important since they constitute the basis of marine ecosystems. They rely on sunlight in order to produce biomass that is then transported throughout the entire food web. Algae blooms can occur if you have enough light available and enough nutrients, and if the water column characteristics are stable enough to allow them to stay close enough to the surface in the layer where there's enough light available. Since the light is decreasing exponentially in the water column, it's actually only the uppermost couple of meters, maybe down to 40-50 meters, where we have enough light for photosynthesis that is big enough to allow algae to develop a larger standing stock. Usually here in the Arctic we have only one major bloom in spring, because after that the nutrients and the surface layers in the water are used up, and there will be no major bloom until the next year when nutrients from deeper layers have been transported up to the surface layers again. In Arctic environments and sea ice covered seas, we usually have algae both growing in the sea ice underneath and algae that are living in the water. When we want to take quantitative algae samples, we just use a bottle that we can lower down in the water and close it at the depth that we want to study, and then bring the water to the surface and filter it on filters that we then use in our investigations. We also add chemicals, fixatives, to these water samples in order to determine, for example, their taxonomic composition. So we do this both by microscopy, that someone is really looking in the microscope on these water samples to see what kind of cells are there and determine morphologically what species we find. But nowadays, of course, we have also molecular methods based on DNA analysis to even identify maybe those groups and species that are too small to identify in a light microscope. We are in addition interested in biochemical properties of these algae. So we are also studying their content of carbon and nitrogen and chlorophyll A. And we are also studying their physiology in order to understand how they cope with the current situation. And we will, in this project, not only do field observations, but we combine our field observations with experimental studies, where we actually take algae material out from the nature to the laboratory and expose them to conditions that we assume to be future Arctic environmental conditions, in order to see how well they can cope and in what sense they would change under those conditions. We have climate models that try to describe how the physical environment in the Arctic is going to change. And to some extent, some of these models are working more or less okay. They have also weaknesses and some points that are badly represented, but overall, the physical future in a way is easier to predict than the future of biological systems, because the coupling between the environment and biological organisms is much more complex and not as easy and straightforward to explain. When we were planning the project, we were aiming at studying two contrasting fjords in Svalbard, one being ice-covered and one being ice-free, and following the seasonal development of these algae. During the course of the project, we became ourselves victims of the rapidly progressing change in conditions, because this winter actually the temperatures have been so exceptionally high that none of the fjords here in Svalbard was ice covered. This has big implications for the ecosystem in the Arctic.